All right, welcome everybody to the last episode of our departmental seminar series this semester. It is my honor to introduce our speaker, Professor Csaba Szabó. I'm just going to do a brief introduction and then we'll hand it over to Csaba. Csaba is a world-leading researcher in pharmacology and experimental medicine, known for his groundbreaking work on oxidative stress, hydrogen sulfide biology, and more recently, cyanide signaling in mammalian systems. He trained in Budapest at the Semmelweis University and completed his postdoctoral work in London with Nobel laureate Sir John Vane, before spending 24 years leading research programs across the United States, including universities in Ohio, Massachusetts, New Jersey, Washington, and Texas. In 2018, he returned to Europe and is currently chair of pharmacology and director of the Department of Oncology, Microbiology, and Immunology at the University of Fribourg in Switzerland. With over 98,000 citations and numerous international awards, his work has had major impact on both academia and drug development. Today, we will have the opportunity to hear him speak twice. In this talk, he will discuss the reproducibility crisis in biomedical sciences, a topic he recently published a book about. And in the afternoon at 4 p.m. at the Life Sciences building, he will share some of his research, some of the research his group is doing on mammalian gaseous transmitters. Please join me in welcoming Professor Csaba Szabó.
Hello. Thank you. I will put the time here just for check. Thanks for having me, and it's a pleasure being here. And I met some old friends, old colleagues, and some new friends. And this is not about science. This is kind of my hobby now, to talk about this stuff: reproducibility, lack of reproducibility, and all the rest. So you will see. And then after this we'll have a question-answer time, and after this you can still stick around and ask me more questions. So whatever you want, happy to answer if I can. So let's see. So the story goes like this. A few years ago I got my first sabbatical, because I worked in the US, and in the US they don't give you sabbaticals, but in Switzerland they do. After 5 years you get half a year off and you can do something else, you can go to another laboratory, you can write a book, you can do something different. And I have some friends at Columbia, and I visited them and we did a little collaboration there. And at one of the dinners we went out to Blue Note, which is this jazz club in New York. And we always talk about science, of course, even when you are at a jazz club. And we always, this question comes up: why is it that we read something in the literature and then we try to reproduce it or try to follow up in our own laboratory, and we have a problem? We have a problem even repeating the main claim that was in that particular paper. And it seems like this is a recurring problem, and it seems like when you talk to people, everybody kind of knows about it. So, and then it turns out that Columbia University has a publishing house called Columbia University Press, and it turns out that they became interested in this topic. And then I was on sabbatical anyway, so okay, so why don't you write a book about it? And so we didn't know if this was actually enough for a book. So the first question was, of course, is it even true, our feeling, that a lot of it is not reproducible? Is it even factually correct? And if it is, then what are the underlying causes? So this is basically the fundamental birth of this story and this book. And and it came out, and I wrote it in English, and then I translated it to Hungarian, and the English version, to my surprise, received some positive reviews. I got asked to do interviews. Wall Street Journal wrote a very nice article about it, and for some couple of weeks it was in the top seller list on Amazon. It didn't last very long, but at least for a few days I felt like I am right next to Carl Sagan, and that was a good feeling. And then Nature put it on the science summer reading list. I figured, they probably felt like if you are too happy under the sun, and then you read this, and then you are maybe not so happy anymore, and that's a good compensation. And then the Hungarian version came out, and also it got some positive reviews. That this Age person wrote that this is some kind of important book, and I got to present it at some meetings. So and it sold out. So now it's actually on the second edition. And of course, the Hungarian Academy of Sciences, they have this periodical called Magyar Tudomány, and of course they wrote a very nice article about it. I am just joking, of course. They didn't do anything. They totally ignored it. So now, this is what we say, or I say in this book. There are at least 5 or 6 claims, and just look at the title of the claims. So, first claim, already controversial. I think that if you look at the whole body of the biomedical literature, then my guesstimation is that at least 90%, if you look at everything that's published, I think at least 90% is not reproducible, not directly reproducible. You go to a laboratory, you try to follow the protocol, you try to do the experiment, at least 90% is not going to reproduce. The second, and I will show you the evidence for that. The second one is that many people say that there are some bad actors, a few bad apples, and this is the problem. It is not, it is a systemic problem. Then we talk about misconduct in the book quite a bit. And my claim there is that misconduct is not due to ignorance or poor training, it's other things, we'll talk about that. Then I talk about the oversight or the lack of oversight. Then I will talk about AI, very, very popular topic. Some people think this is the way out of all of this. I don't think so, but we'll talk about that. I will talk about the publishing industry, which is not an innocent bystander in all of this, but actually it benefits from it, and it kind of maintains it. And I will talk a little bit about how this could be potentially improved or changed, but it would take a systemic effort. So these are the main claims, and let's go one by one. So when we say this 90% number, where does it come from? Well, it comes from articles, direct replication studies that are published in the literature. So this is one number, 93%. This was not yesterday, this was quite a few years ago, a paper published by a group at Bayer pharma company, and they said, guys, we just can't replicate most of what you are publishing. And that was the number that they came up with, 5 successful replications from 67 papers that were published in the literature. Shortly after, another thing came out with this number, 89% was not reproduced. This was a paper in Nature by a group at Amgen. So this was cancer research specific things. The first one was more of a mixture, cardiovascular cancer, cell biology, different things. So these are the numbers. And when I talk about replication, actually there is 2 different kinds of replication. The one that I typically call replication is the direct replication. Direct experimental replication, you read the methods, you set up the experiment, you do everything the way the paper says they did, and you try to get the same answer. There are also conceptual replications and other ways of doing it, but I'm talking about direct replication. I'm not talking about preclinical bench studies, basic research, okay. So this is the table. It's an updated version of what's in the book. And as far as I can tell you, this is the grand total of direct replication studies available today, 5 or 6. This is it. There's no more studies were ever done that would collect a bunch of papers and then try to replicate it in the laboratory and figure out what is replicable and what is not. And when you look at the actual numbers on the right column, the successful replication goes between 7, 11, 13, 17, and 21%. So these are the numbers that are available. There's no other available data, so we have to say that this is what it is. And then if you start to kind of finagle it, and then we say, well, there was a trend in the replication, it wasn't significant, or it replicated on some things but not on others. So if you kind of expand your criteria a little bit, then maybe you end up with like 25% or in some cases a little bit better. But really, if you look at the hardcore numbers, then it's really a low number. Sometimes people say, okay, for example, the null effects were replicated higher. So, for example, 53% of the when somebody says something doesn't have an effect on something, then 50% of that was reproducible. And some people say that's a great result. I think this is terrible. If somebody says nothing happens, and even half of the time even that is not true, I don't think that's such a great achievement. But that's what it is. So these are the numbers, and remember, these are typically top papers in top journals. So this is not the overall field. This is already selected. That's one. The other is that in most of these studies, except the first two, the replicators actually had help. So they went to the original authors, because many times when you read an article, there's not enough information to exactly know how to do it. So they went to the original authors of the paper. They asked for more information which was not in the paper, and using that information, even with that extra, this is the replication that they were able to come up with. So imagine what the replication rate would be if this extra help didn't exist. And by the way, there were also many original authors who were not very helpful in this respect. So they didn't actually give them the information that they were asking for, and then they couldn't even start the replication, and of course that's not in this table. So this story is starting to go out, but it's actually, if you go back to the literature, there is already claims or statements like this going back at least 20 years. So there was this very famous paper of John Ioannidis, who looked at only the sort of the statistical considerations underlying replication. And it's a very complicated paper, and I just want to show you one table that is kind of interesting. So in this table, they calculate the p-value, statistical significance of the original study, and then they calculate what is the probability that using the same sample, the same data set, a second study would also give the statistical significance. And the story is that if the original study has a p-value of 0.05, I can point with this thing, but in the middle one, p 0.05, there's actually only a 50% probability that using the same data set, the second study would also show a p less than 0.05. This is not so important for the laboratory replications, because in those cases, most of what was supposed to be replicated had a much higher significance. So it was more like 0.001, and it was not just one time, but several times in the paper. A main claim is usually not just once, but it's actually repeated through the whole article. So it's not really relevant for preclinical work and preclinical replications. But this, on the other hand, is very interesting for clinical trials. Because when you do a clinical trial with one particular endpoint, and in a phase 2 trial something is significant at that level, that doesn't mean that if you repeat it, it's going to be also significant, just based on pure statistical considerations. So this is just something to keep in mind. But clearly there is something else going on, because even if you have like a p.001, 80% of the time you are supposed to be able to replicate it based on statistics, and instead what we find is that 80% does not replicate, at least. So something is really not right here. Now, people started to think about what this means in terms of costs. This is how much money is probably wasted if you consider a 50% replication rate, which I think is very optimistic. So there's a lot of money that is wasted. I'm going to skip this. And so I think if you, even with the top journals and even with the extra help, you can only replicate this much, then imagine what happens if you start to take random papers published in PubMed and try to replicate those. It's going to be even worse. And so I think the 90% is probably the correct number. And if you don't believe me, believe this guy who has spent his life on reproducibility issues. This is his opinion too. He stated this several times. At least 90% he thinks is not reproducible. And if you still don't believe me, then try to do something. Try to find a paper that you think is interesting and take it to the lab, and then try to replicate it with your own hands. See what you get. But I tell you a few things. So you will have a very hard time finding money for it because it's very hard to get money, grant money for replication studies. Second thing is, the original authors might or might not answer even your emails. Then the next thing is that once you're finished, it will be very hard to publish it. If you replicate it successfully, that's why. If you didn't replicate it, that's why. So in both cases, people will tell you that this is not interesting and this is not worth publishing. And in the end, you put it on your CV and nobody will be impressed because you didn't come up with an original idea, you're just trying to replicate. So these are the sort of the reasons why there are so few replication studies in the literature. It's very hard to get money for it, it's very hard to do it, and in the end, it's not a very rewarding thing in the eyes of your colleagues. Okay, the second statement or claim. This is really not just a few bad actors. And in the book, I put together this kind of misconduct meter. And on the left, I list things that are sort of common problems or issues in experimentation that we are all aware of, but they increase variability, they decrease the chance of replication, and then we go into questionable practice, unacceptable practice, and then all the way to fraud and crime. So it's a sort of a scale that goes from common things to sort of borderline things to bad things. And if you just stay with the sort of common things, and I wrote a lot about this, there are simple things like the quality of the water that can make a difference in certain experiments. Just if you stay with cell culture, here's a random list of things that can affect small details in your experimentation, or problems with misidentified cell lines, genetic drift, contaminations, and so forth. All of these, even what kind of plasticware you use, all of these things can affect your responses. People who do experiments know this very well. But when you replicate, then it's very hard to know what were the exact conditions of the original paper, and then what is it that you exactly try to replicate. Then the reagent is another thing. The reagent quality can differ, antibodies and so forth, impurities and so forth, validation of different reagents or kits. So there is a lot of things that can affect replicability that are nothing to do with fraud. It's just something to do with the complexity of the experiment. Animal models, same story. Everything can have an effect. What kind of cages you use, what kind of animal house you use, what is the flora of the animal house. All of these things. I wrote a lot about this, and we can talk about this more, but clearly, even from the same vendor, depending on, I wrote this also, even in some cases, the same vendor, if they supply you the same strain of animals, if it comes from a different location of the same company, it might even have an effect. So, it's amazing. The handler, who is the animal handler, that can affect it. And lots of other things. So I'm not going to spend too much time on this. Everybody knows about this, and it's very, very hard to keep all of these parameters exactly the same during a replication study. Then there is also these execution variables that can have an effect, and some of them are common things, like people use underpowered studies, people use different ways of validating a certain assay, and there's also lots of honest mistakes that can happen. People are mislabeling something, misreading something, some detail is missing from the protocol, software is corrupted, calculation errors, and so forth. So all of this is still within the sort of normal laboratory work, but it can certainly affect replication. And then when we go into sort of the less acceptable but still common things, then statistical issues come to play. For example, the famous p-hacking, the famous harking. So when people are looking at different statistical tests in order to find the magic significance, or coming up with the hypothesis after the data are already collected. And there's a lot of literature on this. I'm not even going to spend a lot of time on it. But definitely it will affect the reproducibility. And then there is also this other side of the story on the right side, which is really fraud, misconduct. And people don't like to talk about that, especially. But this is also happening, and I have a few slides on that. And so when I got to America, this was kind of the first one of the first things that I didn't quite understand. We were walking in the lab with somebody, an American colleague of mine, and the guy says, you know, these guys here, they are painting mice. And I said, okay, you know, they are painting mice, what does that mean? And, you know, not this, okay, and not this. So what does it mean? Painting mice is an old expression that comes from an actual person in New York who was doing skin transplants, and the skin transplants didn't work because the idea he had was bad. He was thinking that if you take the transplantable organ from a non-compatible animal and then you put it in some magic fluid and then you transplant it, then it should stick and it shouldn't reject. And it didn't work because that doesn't work. But he was actually painting mice onto that color, and then the laboratory technician noticed it, and it was a big problem, and then there was a book about it. And so this is where this thing comes, painting mice, okay? And these people were all painting mice at some point, you know, not in a real sense, but in sort of metaphorical sense. And in the book, I write a lot about these cases. You know, all of them did some kind of data manipulation, fraud, massaging of data, and everyone is an interesting story, and everyone is a half-an-hour lecture. But what is interesting from this is one of the things that's I think is worth noting is that none of these people actually got seriously penalized. Some of them lost their jobs, some of them retired, and there was only one person who actually went to jail. So for preclinical fraud, it's very, very rare that somebody would actually get serious penalty. I don't know any of them actually have to even pay back anything personally. But you know, when you look at the patterns in these misconduct and fraud cases, there is a few things that are kind of common. One of them is that the institutions are very, very slow to react. Another thing is that the wasted money will never recover. Another thing is that the penalties are very, very mild. And in a lot of cases, even if a whole field started out from a bad idea or from an irreproducible or fraudulent thing, a lot of the times these fields take their own life and they continue even after the original finding was already disproven. And this can happen, and it is wasting a lot of money, you know, continuously. So the money wasted is much more than just the actual experiments in these fields. There is more money wasted subsequently. I'll show you a few examples of what I mean by, you know, manipulation. I'm sure that most of you have seen this already. You know, one very common thing is the Western blotting manipulation, where people take the same loading control or same sequence of blots and the exact same, and of course they are like fingerprints, so not everyone is different. And if you take the same thing and you call it something else in a different part of the paper or in another paper, then obviously that cannot happen. The same cluster of cells cannot mean two different things. It cannot you know, that cluster of cells cannot be control, and it cannot be also some other group. So these are flow cytometry. People are using this a lot. You know, you take a cluster of cells, copy and paste it with Photoshop. A lot of these things, this type of manipulations, increased after Adobe Photoshop was invented. So when you look at the curve, as soon as Adobe Photoshop was available, these kinds of things started to appear in the literature. These things are called Frankenblots because they take little pieces and put it together like this. These are, again, the same loading control everywhere. This reminds me to this, I used to play this when I was in high school, you know, this space invader, look at these little things. Then really primitive manipulations, like in this particular story, they actually magnified that little neurite, and they said the neurite grew. The problem is that the background also grew. You know, those impurities also grew, and then, you know, this is the typical thing when the same cluster of some part of the blot is repeated several times, and it's called something else, or when the same histological picture is getting shrunk and rotated and modified, and then called something different. Or when like half a mouse dies, because when you look at the number of animals, then it cannot be that you know, that kind of survival curve is correct. So there is all these impossibilities that are actually fairly easy to detect. And because these are easy to detect and easy to confront the original authors, these are what most of the data detectives, the sleuths, and I will talk about sleuths, look at, because these are easy things to detect. That doesn't mean that these are the only things that happen. It just means that these are the really, really obvious ways of catching problems. And then always the question is, okay, if there's an article which has one component of this, how much can you trust the rest of the article? Because bar graphs or other types of data are much harder to prove because the original data, especially going back, are not typically available. So in this case, for example, Dana-Farber Institute had a lot of manipulations, and they actually had to pay a huge fine recently. This person at the NIH had hundreds of papers that contained manipulated images. He is no longer working there. So there is big stories like this. You can look at this book, for example, just in the field of Alzheimer. There is many problems and sort of misleading the entire field because of foundationally incorrect and manipulated data. So there is a lot of this. And if you say, okay, but these are just outliers. The overall science is correct, and these are just some outliers, and these are just some rogue people. Well, okay. Now, for example, look at this. This is a very recent analysis, and this is looking at image-related problems. So this is not just any kind of problem, there's probably more, but this is just the image problems in studies related to subarachnoid hemorrhage, so one form of stroke. Animal studies involving stroke. As you see, the number of papers increases dramatically over the last 10 years. And the green is the articles where the person didn't find any image-related problems, and the red is the articles where the person did find duplication, multiplication, manipulation. So about half of it, or 40%, already contains problematic data. So his conclusion was that 40% of the papers are probably fraudulent. My conclusion is not this. My conclusion is that, first of all, there is absolutely no reason why there should be so much increase in subarachnoid hemorrhage. There is not more money for it. There is no any easier way of doing it. There is no more groups working on it. So all of the extra stuff above the red line, I think, is not correct. It's just made up stuff. And my feeling is that from the made up stuff, some of them contain easily detectable image manipulation, and the other part didn't contain easily detectable image manipulation. But my feeling is, my suspicion is that's also made up stuff, because there's no way there can be so many studies recently in this field. It's just no way. Okay, so some people say there's this tip of the iceberg analogy, that these are just sort of, a few bad apples. I don't think this is the case. In the book I use a different analogy. I call it like a swamp, and there are all these swamp creatures thriving in it. And one thing I want to emphasize is that in the current system, when like sleuths, data detectives are looking for image problems randomly almost, and only looking at image problems and not at any kind of other problems, only the amateurs get caught. So currently, if somebody is doing sort of the lazy and bad job of duplicating or manipulating images, those get caught. But my feeling is that there is a lot more that's basically better fakery that is harder to catch, and those are not caught in the current system. In the book, I have a little whistleblower account which was published on a blog called Retraction Watch by somebody who was working in one of these laboratories that was involved in data manipulation. And this person, probably he or she, explains how that particular laboratory worked and how there was these different ways of controlling information, that there was the sort of the underlings who were producing data, but nobody knew what the other person was working on, and all of this data ended up in these middle managers, and these middle managers were the ones who were putting together the figures, but nobody knew exactly how those were put together, and they were presented to the top boss the way the top boss wanted the data, but the top boss never actually did the manipulation himself, so he was kind of protected from this. So this is just a kind of a AI-generated explanation of how this works. And I'm not saying this is the only way these laboratories can work, but probably this is one of the ways how this can happen. And there is also in this blog a lot of information on what kind of pressures were applied on the people in the silos to actually produce the correct data and so forth. Okay, we could talk about this more, but so this brings me to the next story, which is that misconduct is not just, or no, mainly not about ignorance or poor training. It's about pressures, perverse incentives, weak enforcement, and these are some of the contributing factors: structural, organizational, cultural, individual. And I show you a few numbers on this. So this is one number. This is 10%. This is the lifetime chance of a PhD student ending up in any type of academic position. American numbers, but I don't think it's better anywhere else. This is another number, 0.5. This is the lifetime chance of a PhD student becoming a tenured full professor any time in his or her life. This is the number of PhDs that are being churned out from the system, and that is the tenure track positions that are not changing over years. So there is this gap increasing. When you look at young graduates already moving into the field of biomedical or engineering or STEM research, they already have a sort of lingering feeling that there is cheating and there is unfair competition. So they already feel this early on. This is a very interesting number. This is from an anonymous survey at the University of California. Again, biomedical trainees, very, very young people. 80% said in this survey that they are willing to massage or manipulate data in order to get a grant or in order to get an advantage over the other person, 81%. This is zero. This is, for example, the amount of money I got, or the amount of money most people get from their institutions in the USA in order to run their laboratory. Everything has to be brought in from the outside, okay? So they maybe give you a startup fund, and then eventually they expect you to bring in several grants in parallel all the time in order to maintain the laboratory. So here is the laboratory that we had in Texas. On the right, you see the lights. That's the only lights because this is only our group was working when I took the picture. And there were other groups in the building too, but yeah. And this big monstrosity, this was built while, in a few years, because this is the clinical. In America, the clinical is bringing money, typically. The research is costing money. So, they build more clinical stuff, but the research is sort of something. If you want to be a researcher, you have to bring in all the money from the outside. And over the years, what happened is, again, USA, that the money that went into research, the grant money increased, but because the grant money increased, all the institutions increased their capacity for biomedical researchers. So in the end, the net result was that the person funding actually decreased because there were way more people trying to get money than the amount of increase that happened. And then eventually the amount of increase stopped, but the people were still there asking for money. So, what that ended up doing is actually it decreased the chance of everybody getting funded. In the US, there is also this thing called overhead, which is really driving a lot of this. Meaning, in many places, if you bring in $1 million of a grant, your institution gets another 700,000, 800,000, sometimes even a million, which is going for administration. So for a university, to have somebody who brings in grants is a very, very lucrative business. They bring in all this overhead extra. So obviously, there is a pressure on everybody to bring in this. And that brings us to the grants game, which is another story. This is kind of the current rejection rate at the NIH for a typical individual grant. This is the rejection rate currently in the Swiss system for a young person starting grant. This is the rejection rate of an ARC currently. This is the number of revisions you can do at the NIH. This is the number of revisions you can do in Switzerland. This is what these study sections look like. I have been spending a lot of time in these study sections. This is where people get together and make an evaluation of the grant and vote and rank the applications. This is what it looks like. This is what it feels like. And this is the sort of the graph that shows that the young investigators are a sort of an extinct species. So currently, I think it is 40. So the average age when in the US system a young investigator gets his or her first independent grant is 40 years old. So it takes so long to even start your career because of all these grant issues. And of course, you have to always push for novelty, but you also have to guarantee the novelty, and you have to also guarantee the success, which is impossible. And there's all these things that are not welcome, repurposing, confirmation, replication. These are not things that you can ever really get significant money for. And here is one other interesting number. This number is what is called the inter-rater agreement of different reviewers, meaning that if you have several reviewers, they typically have widely different opinions about the project. And in the current system, when it's so easy to get a rejection, it's enough really to have one who has a really negative opinion, or even an average opinion. If the others are good, even that one is enough to kind of kill the project. And this was very well documented, these inter-rater agreements. Nobody likes to talk about it. There were different studies. These are the studies showing different inter-rater agreements. All of them are really bad. What should be a good number is 0.75. It is never that much. It's not even close. There's also a lot of discussion. Recently, I read this also in Hungary. Whistleblowers were saying that there is politics that is overruling. So even after somebody would actually get a good score, even then, sometimes there are other considerations, political manipulations that sort of change the ranking and push in grants that were not in the selected ones, and vice versa. And you don't need actually whistleblowers to know this. All you have to do is, like in the book, I have this graph, which is a distribution of the grants that were funded, the grants that were not funded, and the percentile. And you can see that, for example, even though, let's say, the pay line is 10 percentile, you can still see that a bunch of grants got funded even in the 20th and 30th and 40th percentile. So those are grants that were not supposed to get funded, but they still got funded. Somehow they were pushed in to the funding level. You can also see that even in the 5th or 6th or 7th or 8th or 9th or 10th percentile, some grants didn't get funded. So something happened there also. So without even having a whistleblower, you already know that the pay line is not an absolute. The pay line is just some kind of recommendation, but these grants administrators, due to all kinds of other points of view, other considerations, can actually change who gets funded and who doesn't. Okay, oversight. That's another long story, and I'm going to go faster on this. But basically, with any kind of crime, any kind of misconduct, you have to balance two things, the cost of crime and the rewards of crime. So the cost of crime is related to the probability of detection and the severity of the punishment. So people had these discussions, which is more effective if you have a higher probability of detection or if you have a higher severity of punishment. And there's different opinions about this. But if you, like in the current system, my point is that both the probability of detection is low and the severity of punishment is also low, and at the same time the rewards are very high. So everything in this balance is kind of misaligned, and that's a real problem. Because who is really involved in looking at misconduct or looking at problems in a publication? These are what you would think should be the people or institutions who would do it. The investigators themselves, maybe their institutions, maybe the funding agencies, maybe some kind of science ministry, or maybe the journals or the publishers. None of them are looking at anything. Once it's out, it's out. Nobody goes and starts to look at, is there any problem there? Who is looking? These guys. These guys are what are called scientific sleuths. Many of them are ex-scientists or retired, and these have it as a hobby, as unpaid volunteers, to look at the literature and find and identify image manipulations, data manipulations, problems of different sort, and then disclose it and hope that eventually the journal will retract it and eventually something will happen. And these are the guys who are currently doing the scientific integrity for everybody, and guys mostly, and girls. So these science sleuths are sometimes sued, sometimes threatened, sometimes taking advantage of, meaning that they find something, they post it on PubPeer, which is this online forum where people can post problems of this type, and then the sort of the legacy journals read the stuff in PubPeer and then write these exposure reports. That they forgot to mention that these were actually discovered by the sleuths. So it's kind of not an ideal system. Sometimes some of them get awards, sometimes some of them even get cash prizes for what they did, but most of them really, and okay, this guy got 2 million exposing the Dana-Farber problems, but most of them doing it basically for free in their free time, and they don't actually get any compensation for it. But at the same time, they are still exposed to potential lawsuits, potential threats, potential career assassinations, and all the rest. There are some bright spots. I already mentioned, I already mentioned PubPeer. There's a plugin that you can put on PubMed. That's a PubPeer plugin, and if you find an article, this will alert you if that paper has comments in PubPeer, and then you can look at what are the problems. It will tell you if this is potentially a problematic paper. Retraction Watch is out there. This is another website. It's not proactive, so they don't actually discover anything, but if somebody had a retraction, then they write about it, and then you know that something was retracted, and you can look at the broader picture. There is this one, it's one of my favorite websites, it's called For Better Science. Leonid Schneider, an ex-scientist, is running it. They actually expose new problems on a weekly basis. This is the likelihood of detection, one particular number. This is the percentage of problematic data found in NIH grant applications, meaning that the level of detection is so low that this is not going to have any kind of deterrent value. I'm going to skip this. I'm going to skip this. Okay, AI. Let's talk about AI. Everybody likes to talk about AI. AI. With AI, one of the things you can do, you can create beautiful new images that look like the real ones. All you have to do is train it on actual images and tell it that you want it to be a little bit inflamed or a little bit, necrotic or whatever. It will create an image that will look exactly like the real thing, and it's going to be original. It's not going to be anymore a copy of something before. So it's no longer the thing where somebody copy and pasted something from an old paper, relabeled it, called it something else, and you can detect it. These AI-generated images are very, very hard to detect because they are original. They are not anymore copies of something else. And people started using this in these paper mills and sell it for paying authors. I will talk about paper mills. Another AI stuff happening recently is that there are all these public databases for clinical data, drug responses, all kinds of things. Publicly available open source databases. Everybody thought this is a good idea, of course. Open source, open science, we love it. What happened was that some paper mills figured out a way to extract data from these open source databases and then organize it and analyze it according to some criteria and write these synthetic papers by the thousands and then sell it. And these are, spurious relationships, correlations that will never really replicate or validate because they are just basically sucked out of data and over-analyzed. Thousands, tens of thousands of papers being sold like this. AI. Here's a recent paper looking at how the paper mill products, a lot of them AI generated, are increasing over the years. This is the number of likely problematic papers being posted even on open servers like Archive and bioRxiv. It keeps increasing, so people are posting these things already. Sometimes, smaller things like people put messages that AI can only read because they are in white font, instructing AI to be a favorable reviewer of your CV or paper or whatever. This is small and kind of amateurish, but this is also happening. There's recently a big outcry on something that I think is relatively minor, that a lot of papers now start to contain hallucinated references, meaning, because if you ask AI to come up with something, a lot of the times it actually comes up with references that look like real, but they are not there. They just makes them up. So people are kind of starting to lose their composure over this, when I actually think this is a relatively small problem compared to all the rest that I was talking about. But this is now, in the literature. I actually wrote a LinkedIn post. Sometimes I post some things on this LinkedIn, and I had AI actually write a cartoon about this. So these are all the people who are getting all out of shape for AI-generated references, when all these other problems are prevalent in the literature. People seem to like it. It's like a lot of reposts happen, and people seem to like this for some reason. Now, some people say that AI will eventually anyway take over. So we are in a transitional phase. All this paper stuff is not going to happen in the future. It's going to be either papers written already and presented for AI, and eventually AI will anyway take over all aspects of science, hypothesis generation, data analysis, everything else. So I don't know, but there is certainly a new way of thinking like this when AI is going to do everything in the lab for you. Including generating nice cartoons like this. Finally, the publishing industry. Okay, so we can talk about them too. The publishing industry like to pretend like they are victims in this, or that people are just we are just publishing what people are sending to us, and it's nothing to do with us. But actually, it's a for-profit system for the most part. So they will make money if there is more papers coming out. They will make money if people pay the open access fees. They will make money if they publish more. This is clear. And the profits of the publishers are enormous. So, for example, just compare the profits of the publishing industry with the profits of sort of more, regular industries, banks or car companies or supermarkets. So it's a huge, huge profit margin that they are incurring. So no surprise that the number of journals is increasing. No surprise that the number of papers is increasing. And I don't think the money that goes into research is increasing, so it is just extra stuff that somehow appears. And of course, this happens at the level of the credible journals, and what happens now is that because these credible journals are also getting so many submissions that they are creating these next year sort of baby journals, where they refer your submission. If they don't like it so much, then it's okay, send it to the next level or to the next level. So they are multiplying all these journals, and on top of it, you have these predatory journals that will basically almost just publish anything. So there is all these places, all this room for material to be published, and my fear is that there is more room than actual material that could fill it. So then, there's all this other stuff that will fill it, AI-generated stuff, fake stuff, paper mill stuff. And there's lots of problems with the publishing industry. One of them, this is actually Hungarian people figuring this out. It's not that hard to figure it out, actually. How much money are we donating to the publishing industry by refereeing for free? We are all refereeing for free. And why are we doing this when the publishing industry is making all this profit? It's a mystery to me. But it's billions of dollars each year that we are donating to the publishers. So this is a nice cartoon. So the publisher says, you will write papers for us, you will pay for us to publish them, you will review these papers for free, and you will also pay us to read those papers. And then the guy says, I changed it so that it's not crude, I won't do this. So he will just not do it. But most of them will do it. Most of them stay in the system, and this is the system currently. And why all of this happening? This is the number of publications in the dark. Number of publications going up. Look at this. This is unbelievable. And then, if you calculate what is actually the value, the disruptiveness of each of these papers, that goes down like crazy. So each paper is less and less valuable, but there is much, much, much more of them. And if you look at what is actually in the papers and how the papers are written, the hedging is decreasing, the sensational language is increasing. So all of this is changing and not to the benefit of anybody. And of course, there is all these paper mills, all these fraudulent papers that are basically units of publication for sale, prepared by groups which have nothing to do with science, and they just make up data and make up stories, and they sell it online. And according to some estimations, in some fields, the percentage of fakes published is 20-30%. I say it one more time. 20-30% of what you read is already fake straight from the outset. Fake. Nothing to do with laboratory, nothing to do with experiments. Just sold and bought. And so these are the millers, not that miller, the paper millers. And the paper millers make this much money each year. This is the amount of the paper mill industry each year. Average is 300 million, 3,000 million, 3,000 million. It's a lot of money. And you can find them on online. They advertise. Some of them say, we are not cheap because we guarantee publication and indexing by 100%. And then you go online and you like whatever your topic you like, and there's the title of the article, and then you can buy, authorship for how much money, first author, last author. They give you special deals if you buy the first and last, then it's a better package. They don't tell you what the journal is, but after you buy it, then you will know that. And of course, they arrange for a favorable refereeing, and then they arrange for the publication of the paper. And none of this is real. None of this has any real data. This is all made up on somebody's computer. Now, somebody actually went into the trouble and did a very elegant network analysis on how these papers actually end up in the literature, because it's not enough to make it, produce it. It's not even enough to buy it. These paper mills have to have a way to actually get them in the literature. And some of them are so primitive that anyone who looks at them would immediately tell that this is not real and we shouldn't publish them. And yet they are getting published, meaning there has to be somebody in the journal's editorial system who lets these things through. So these guys actually did very elegant network analysis and identified checkpoints, meaning individual people in different journals who are doing this sort of letting through and pushing these paper mill products into the system. And I did my little sting operation a few years ago, because I get these inquiries from these fishy places that we would like your editorial help, we would like you to help with publishing something. So one time I said, okay, well, tell me how you want me to do this. So they started to send me all this information, and in the end I wrote it up, and then it was published on a blog, and then it was covered by Science. And so first thing, they switch immediately to WhatsApp, okay? Because for some reason that's better than your official email. Then on WhatsApp, they start to send you the information, like how much they would pay you if you push one of their papers into the literature. They will, at one point I asked them, okay, are you sure, how can I be sure that you are actually paying? So they were foolish enough to actually send me a list of other people that they actually paid for this type of service. So now I have that too. And then they send me eventually the price list. So look at this. So if you get one of their papers into a sort of high impact journal, they pay you 16,000, 12,000. So not a little money. I mean, this is quite a bit of money for each paper that you push into the system for them. So if somebody takes up on this, that is a very, very lucrative business. I mean, imagine this. Imagine how much money is flowing through the system like that. And it never ends. They keep sending me these things. This one is like a price list of what kind of topics they want, and I mean, it's just unbelievable. And then retractions are happening, because eventually some of these things will get retracted. Not all of it, most of it is probably still out there, but some of it eventually gets retracted, and the number of retractions is increasing, but again, it's only the tip of the iceberg. There's much more problematic stuff on paper than retractions. I would say probably 100 times more. So, meaning most of it is out there and it's not retracted. Okay, and then the last thing is what to do. And I don't know if I have enough time to explain all of this, but I will just give you some idea. So there's plenty of bad ideas out there. Okay, so that's one thing. If you look at what people are writing about, this was in Nature. Here. How to spot when a paper is fraud. This is what Nature is advising. Pay attention to the science. Imagine, this is why you pay Nature for. So they tell you that you should pay attention for the science. Amazing. Then, this is another great idea. Somebody wants to have like a nutritional label on papers, like on food, right? Okay. Then another idea, ISO 9001, sort of like a quality control, but who is going to enforce it? I mean, these journals are going to, of course, investigate themselves, and they will find themselves perfect. Then some people want to have like an external evaluation system for journals. Who is going to do that? Some people want to give researchers a lifetime word limit. Who is going to enforce that? Some geniuses want to have these token systems, so that, because another thing that happens is that it's getting harder and harder to find reviewers. Nobody wants to review anymore. So they want to have the system where if you review, then that will give you some extra credit so that your stuff also gets reviewed faster. So it's like a going back and forth. without discussing the elephant in the room, which is that all of this is done for free, and publishing industry has plenty of money. So, researcher verification indexes, some people want to come up with that, so that you know who is a real researcher and who is not a real researcher, I don't know. So there are plenty of bad ideas. What about good ideas? Those are mine, of course. So I wrote a whole bunch of stuff about this in the book, and basically, it's not even my idea. This guy, Doug Altman, said it many, many years ago. I think this is all true. We need less research, better research, and research done for the right reasons, okay? So those are really, really strong principles that I think are all true. Now, what is the usual suggestions? What is what people do when you go to these integrity courses, when you look at guidelines, manifestos, and whatnot? They usually talk about better training, blinding, randomization, pre-registration of the studies, and they talk about funding replication studies. Nobody funds them, but they talk about it. And these things are usually embedded in these guidelines, which every country, every organization has, and including Hungarian Academy of Sciences. I just put this up there today. You can look it up. It's all there. It's all very, very neat and elegant, but broad, generic, voluntary, non-binding, no consequences if nobody is checking if anybody complies, and nobody gives any consequences if somebody doesn't comply. So in the end, it's a nice piece of paper and has some nice principles, but this is not really working. Latest one is this thing called Stockholm Declaration, and what the Stockholm Declaration says is that the way to make ahead is to have more journals going into scientific societies, and also to have more of these diamond open access type access to journals. So it's still going within the existing publication system, but if you look at what the society-owned journals look like versus the sort of industry-owned journals look like, there's not that much difference. They are both charging very high access fees. They are all sort of working within the same framework. So I don't think this will make a difference. People are making enormous amounts of money on all of these, even the sort of non-profit publishing industries. So, these are still looking to me like for-profit. It's just they are called non-profit or not for-profit. Like secretary and CEO is making about $900,000 at the American Association of Advancement for Science. So I wrote my counter thing, which is why the Stockholm Declaration will never work, because I don't think it will ever work. So this is coming out now. But anyway, so basically, and we can talk about this after I finish, but I think all of this should be somehow totally reorganized and rethought. The way we are training scientists, the way the funding agencies function, the way the institutions interface with them, and the scientific publishing model too. All of this should be done differently. And I tell you one more thing. If somebody would start this whole thing and set it up from scratch, nobody would set it up like this. This is not how any logical person would set it up. With a for-profit publishing industry that is making billions out of public money, who would do that? To have a system where nobody is checking if what is printed is true or replicable, who would do that? So it just happened. It evolved, if you can call that evolution, it kind of developed into this, and this is what we are dealing with. So I have some ideas on how the training should be changed with a focus on reproducibility, maybe even like a separate track of training, which would be not mainly focusing on doing science, but actually checking science, replicating, reproducing, analyzing, and these people could be hired and work together in a team, or could be used in granting agencies or journals. There should be a totally different way that the academic institutions should look at this. They shouldn't look at it like research is bringing money. They should look at it like, yes, research is costing money, and they need to invest in some research from their own funds, especially in America, where you have these huge endowments which are just sitting there and not being used. I think the big change should really be the granting agencies. Right now, granting agencies are basically places where you apply and they give you the money, and God bless you, do what you want, and eventually send me a report that nobody will read. I think the granting agencies have the power to change all of this. And if the granting agencies would become more like partners and collaborators, and they would actually be involved in the research process, not just giving the money, that could make a huge difference. They could build in replication, they could build in verification, and they could help the institutions to establish quality control systems. And that I think would make a huge difference. And the publication industry, basically, I think, should be basically put in the hands of the founders. The funding agencies could run their own journals, and they could just mandate that if I give you money, you do the research, eventually you upload all the data, we'll store it, we'll make it open access if it's like that, and then we'll publish your paper online, and we'll have post-publication review, and all of this for-profit industry would basically stop. If the granting agencies would control the dissemination, then what would the public agency, for-profit agency do? Nothing. So I wrote a declaration on that, and basically, in this alternative system, basically the granting agency would be a partner with the researchers, and eventually they would put the stuff out, they would arrange a replication, a post-publication review, and then it would be like sort of like a living document. So after somebody does something in relationship to the first one, they could post it, they could edit, they could make comments on it. So it would not be like a document set in stone, but it would be more like a part of a process. And general public should need to know about this more. But basically, this is the thing. So the funding agencies, I think, could really, really make a difference. Because in the end, they are managing public money. And the public money should be used to the benefit of everybody. And it shouldn't be used in a system where 90% is not reproducible, and it shouldn't be used in a system where a lot of money is flowing out into the for-profit publishing industry. This makes no sense to anybody. At least not to the taxpayers, not even to the scientists. Imagine all the money would stay and use for science instead of paying for open access fees and all the rest. So when I put the whole situation into AI, then it gives me this kind of picture, which is a bit really depressing. So there are all these idealistic people in the granting agency, there is this establishment doing the shady stuff, and then there is this meta-science people who are trying to improve, but they are really just bozos and kind of, riding in the back but not making any difference. I think it's kind of neat or depressing. And so, I mean, this is it. I mean, I think I kind of explained what I want to explain. We need research integrity and science reform. And then when I say it, then nobody likes it. The granting agencies don't like it. The publishers don't like it. Nobody likes it. But honestly, I don't think this can go on for a long like this. This is all the trends that we are seeing, or the trends in the paper mills, the trends in the AI, the amount of money being sucked out of the system, the funding rates decreasing, the difference between how many people we train versus what the available jobs. All of this is, I think, looking in the wrong direction or pointing to the wrong direction. So something has to be changed, and maybe these are not the best ideas, but somebody should come up with something, because I think we are losing a lot of young people who are not choosing science anymore, or choosing science and very quickly disillusioned, and then they don't continue. And the quality of what we are putting out is much worse than we would like everybody to believe. So people will eventually wake up to this and they will say, why are we funding all of this if it's so irreproducible? So there has to be some change, okay? And this is, I think, there's one more slide here. So you can read some things about this. The science fiction's book is a good book, mainly on statistical things, but definitely a good one. The Charles Piller book about Alzheimer is a really good one. The rigor mortis is also looking at the economics of all of this and all the money wasted. This is not directly related, but one of my favorite books is Free Radical about St. Georgi. So there's a bunch of books that are probably worth reading.